Welcome, everybody, to the first session of the 2026 year. This is the 24th year of a course in bridge building, which is called Demystifying Medicine, and it comes to you from the National Institutes of Health in Bethesda, Maryland. The goal of this course has been to bridge the exciting developments in biology, engineering, and computer science with medicine. It is not last week's research seminar. Its goal is to excite you, no matter what your background and discipline is, and therefore it's welcomed by students, fellows, postdocs, PIs, program directors, etc. The course is going to be entirely virtual this year, and it's held on Tuesdays from 3 to 5 p.m. Eastern Standard Time from today until the end of May. The link for you to watch on, which is what you're on now, is listed here. And for those who seek CME credit, the CME code for today's session is 67264. Now, most importantly, we urge you to submit questions during the course of today's presentations. And you can do this by sending it by clicking on your Send Live Feedback on your videocast display. These questions will then be examined, and we will try to get to as many of them as possible in a session which follows each of the two presentations. Now, I would remind you that all previous sessions from the very beginning, 24 years ago, are archived, and the link is stated here, and we urge you to access these at any time. If I have any other request any other information, please get in touch with us through the link that's listed on the slide. So the entire purpose of this course is to bridge silos of information. That is, where people know a great deal about their own discipline, their own topic, but they know next to nothing about what's in the silo next door. So our logo is the Brooklyn Bridge, which connects Manhattan and Brooklyn. And we are, as always, like the individuals on the catwalk, sharing information coming from different disciplines, both translational, clinical, and basic, in the attempt to build exciting bridges which will encourage better research and advance progress for human health. Now, next slide, please. So today's topic is certainly one of the most challenging ones in our entire world situation. And that has to do with having survived, I think, 2 pandemics just within the past 30 or 40 years. Are we ready for the next one, whether it be an epidemic or a pandemic? Or will it be another 100 years, like the difference between the so-called Spanish flu epidemic of 1918 and then the emergence of HIV and COVID-19? And will the next pandemic or epidemic be a virus, a bacterium, a fungus perhaps, or even something more exotic, an Andromeda strain, about which, of course, we know nothing? Now, consider in today's presentations some important issues. For one, the world is much smaller because transportation is rapid, massive, and accessible. In 1950, when Franklin Roosevelt dedicated the National Cancer Center and opened up the Bethesda campus of the NIH, he pointed out the need for communication across the globe because it could take 3 days to get from one part of the world to another and transmit an infectious disease. That 3 days has been now reduced to a matter of hours. We're also concerned about the fact that it would seem as if global health programs are perhaps weaker, certainly being challenged, and national public health programs likewise. And then we also have, in the United States, a certain serious problem of scientific denial, including reevaluation of vaccines, and those feelings are abundant. And then there's a third huge, another factor, and that is the effect of global warming, which is changing infectious agents as well as their vector distributions and also susceptibility of hosts. We're going to have a separate session in this course on global warming in another, I think, 4 or 5 weeks. I think the bottom line is that constant research is required because the infectious agents are really the ones in control. They can mutate, they can develop resistance to any therapy that we develop, and they are capable of jumping, for viruses at least, from one species to another, which changes their whole clinical and public health problems. And they kind of select patients, perhaps. In some, the very young, and some, the very old, etc. So this is an enormous problem. And we are really delighted today to have 2 global experts to discuss various aspects of this big question of, are we ready for the next epidemic or pandemic? Next slide, please. Now, our first speaker is going to be Jeffrey Taubenberger. Jeff is a renowned virologist and molecular pathologist who became quite famous in 1996 when he sequenced the genome of the influenza virus, H1N1, which caused the 1918 flu pandemic. And this was in the form of a breakthrough paper in 2005. Recall that in that pandemic, roughly 50 million people died in the world and something like 700,000 in the United States. Jeff got an MD and PhD degree from the Medical College of Virginia, and in 1988, he came to the NCI for training in pathology. And he was one of the first to apply molecular techniques to pathology. And in this, he developed new PCR techniques and others that really set a standard for this kind of investigation. In 2006, he became a senior investigator at the NIH in the Laboratory of Infectious Disease, Viral Pathogenesis, and the Evolution section of the NIAID. And he carried out, and still does, some absolutely astounding influenza research. In 2005, he was appointed the acting director of NIAID, and he has continued in his research and substantial administrative responsibilities. Jeff has received many awards, citations, publications, and leadership, as well as committee activities. I've heard people say that he's the world's leading authority on influenza virus, which will be the major topic of his talk today. Now, our second speaker is Arturo Casadevall, who's an internationally recognized infectious disease expert. He received a degree in biochemistry from NYU, medical degree, did a residency in Bellevue, and then went to the Albert Einstein College of Medicine, where he was trained in infectious disease, and then by a very productive immunology fellowship with Matt Sharp. At Einstein, he became professor of medicine and also remained very active in the infectious disease clinical activities, and was chairman of microbiology and immunology. In 2015, he was recruited to Johns Hopkins as a Bloomberg Distinguished Professor in the School of Public Health and Medicine. He is responsible for outstanding research on fungal pathogenesis and is a leader also in improving doctoral training. As one of his best statements that I've enjoyed is his goal to put the P, philosophy, back into the PhD and other educational programs at all levels. So Arturo has received numerous national and international awards. He's an elected fellow of the National Academy of Medicine, the American Academy of Arts and Sciences. He's had extensive NIH and international advisory responsibilities, extensive research, commentary, publications, and is known globally for the excellence of his work and opinions. So our first speaker will be Jeff Taubenberger, and then he'll be followed by Arturo Casadevall. And please submit your questions during the presentations, and we look forward to a lively discussion at the conclusion. So, Jeff, would you please proceed?
Thanks, Dr. Arias, for that very kind presentation and introduction. It's a great pleasure to be participating again in a Demystifying Medicine talk, and to see you, and to especially to be presenting with Dr. Casadevall today. So, are you seeing my slides?
Yes, we are. Perfect.
Okay, thanks. So, to start off, I have no financial disclosures, and the objectives here on the screen, although I joked this morning with people in my office that I could give you, as in the military you would say, the bottom line up front. So the bottom line of the talk up front is that influenza is bad, both in its epidemic and pandemic forms, that we are woefully inadequate at predicting its future evolution, both whether annual epidemic strains, how it will behave year to year, or the emergence of new pandemics. And I can say that, unfortunately, all the vaccine strategies that have been licensed over the past 80 years to control influenza are inadequate in their control and are suboptimal, and we need to do better. So I was going to say that could be the end of the talk, but if you want me to go on, we'll continue. So we'll see if I can get this to. So. We'll just start with the virus itself, and very quickly, I'm going to show a lot of slides and kind of talk too fast. And if I try to make an important point here and there, I will let you know if it's important. So flu viruses are incredibly diverse genetically and antigenically and ecologically, as you will see. We're going to be talking specifically about influenza A viruses, which are negative sense single-stranded RNA genome viruses, in which their genome is separated into 8 distinct segments of single-stranded RNA, and that each contains at least one, and sometimes 2 or a couple of open reading frames, so that the virus makes about a dozen proteins. It's an envelope virus. It has 2 major surface proteins, hemagglutinin and neuraminidase, which have sort of opposite goals. The hemagglutinin is to bind to target sialic acids on the tips of glycans of the cells it's going to infect, that is in humans or other mammals in the respiratory tract, and in wild birds in the lower GI tract. And the neuraminidase is the sialidase enzyme that actually cleaves the newly formed viruses off as they bud from the viruses to allow them to escape from the cell. There are 2 major features about the virus that are important to know. The first is that it's very sloppy in the way it copies its genome. It doesn't have proofreading like your cells would have. So that it is very error-prone polymerase, so that it induces a lot of mutations. So on average, about 1 every 1,000 bases has a mutation put in it, so that basically every possible mutation is made in a swarm of viruses. So the viruses live sort of in a quasi-species swarm of sort of every mutation possible. And the second big feature is because the genome of the virus is separated into 8 little segments and not on a single string, say like a measles virus, is that if 2 different influenza viruses infect one cell within a person or an animal, you can mix and match these gene segments. As long as you have all 8 represented, you can create 256 different versions of viruses with all the different possible combinations of these gene segments. So the first point mutation change is what has been called antigenic drift or evolutionary drift, and the second one has been called shift, where you have a more dramatic change, but both of them play a huge role in influenza evolution. Another big problem to consider is that while we at the NIH, and for most of you, we are considering influenza as a human problem, and it is certainly a human problem. Humans are probably kind of accidental hosts to influenza viruses. The natural reservoir are a wide variety of over 100, maybe up to a couple hundred species of wild birds, mostly wild waterfowl like ducks and geese, also shorebirds, but other orders of birds as well. The full ecologic range of influenza A infection in wild birds is not known. But the other thing that can happen is that influenza viruses can transmit from their wild bird natural hosts to a range of other animals, both domestic avian as well as wild domestic mammalian hosts, as divergent as pigs, horses, whales, seals, camels, bats, etc. So the influenza viruses are really very remarkable at their ability to adapt to many, many warm-blooded animal species. I think that I like to show this picture as a picture of Darwin about 1860 and a snap from one of his notebooks where he draws out thinking about how things might evolve, this sort of an imaginary or an example of a phylogenetic tree of the genetic and evolutionary relationships between organisms. And I think he would be fascinated by influenza because one can look at the evolution of influenza in terms of years and decades, as opposed to traditional eukaryotic evolution. So its evolution is incredibly rapid, and I think that he would find it, I think, really interesting as a model of evolution. So the classic view then of how viruses get to humans is that you start out with a virus that's shed out of the nether end of a duck. It's an oral fecal transmission in waterfowl, like this duck showing its cloaca, and somehow it goes from the cloacal orifice to the human nose. Whether that is a direct transmission or involves an intermediate host like shown here with this pig is still unclear, although there's clear evidence that humans and pigs do share influenza viruses. I don't know how often it is through this particular mechanism, but this is a possibility. The evidence actually is such that humans give influenza viruses to pigs much more commonly than pigs give flu viruses to humans, although it does happen in both directions. As a pathologist, I always like to give a hat tip to Rudolf Virchow, who was the father of pathology in the mid-19th century. But among the many different career hats that he wore as a well-known public health and epidemiologist as well, he also termed the coin zoonosis, saying that between human and animal medicine there is no dividing line. And I think this is very important, especially when you think of agents like influenza that are not very host adapted, that are very flexible pathogens that can move between hundreds of species. So if we look at what we know about influenza pandemics over the last century plus, we know something genetically about 4 influenza virus pandemics, the oldest being the 1918 so-called Spanish flu that occurred starting in 1918 and into 1919. And that then circulated as seasonal flu until 1957, when it was replaced by a shift event for the so-called Asian pandemic of 1957, which was H2N2 subtype, different hemagglutinin and neuraminidase subtypes. And then 11 years later, another shift occurred where the neuraminidase remained, but a new avian-derived hemagglutinin, in this case H3 subtype, appeared. And then in 1977, the pre-1957 H1N1 virus recirculated again and circulated and co-circulated with the H3 viruses until 2009, when we had another pandemic that was the so-called swine origin H1N1 pandemic, which caused the prior human H1N1 virus to go extinct, but not the H3N2 virus. And now, since 2009, this lineage of H1N1 has co-circulated with H3N2. Of course, we're all familiar with SARS-CoV-2 in 2019, 2020. And then we can go back in time, and there was a so-called Russian flu in 1889, which has long historically thought to be influenza, but some people have recently suggested that it might have been a coronavirus pandemic and possibly containing the ancestor to what is now a cold virus, an endemic human coronavirus, OC43. But without having samples from this time period, it's impossible to answer this question. If you look at the medical records without any biological data, you can suggest, based on the patterns of how unique influenza pandemics look, that there have been any number of, say, around 14 or 15 pandemics in the last 500 years, average interpandemic period of about 35, 36 years without a seeming pattern. They had come as close as just less than a decade apart and as far as about 150 years apart. Going back at least to about 1510, you can perhaps speculate about pandemics prior to that, but it gets more and more difficult to interpret the available medical literature to that extent. But there seems to be no pattern, and while the world population is dramatically increasing, it does not look like the pace of pandemics is directly tied to global population, at least so far. So when we think about future pandemics, we've had 4 pandemics that we know about in the last 100 years, likely 14 pandemics in the last 500 years. What do we know about the future of the next pandemic? And the problem is that we are absolutely incapable of, at this point, in predicting what the next pandemic will be, of influenza, what subtype it will be, where it will emerge, how virulent it will be, how it will behave. And so recently in an exercise, I thought of about 15 or so different ways in which pandemic viruses might appear in humans from different direct transmission from avian to through possible intermediate animal hosts. And the last one, number 15, is release of a prior pandemic virus from a freezer is certainly something that in this day and age we do also need to consider. For example, the 1957 H2N2 pandemic exists. It's not a select agent. It exists in research laboratories and public health laboratories around the world, and H2 viruses have not circulated in humans since 1968, so the majority of the world's population has not seen H2, and so this is a virus that I worry about, and I think that there should be more controls on the control of where these vials are and how they are stored. So I want to talk about a possible pandemic threat that has been in the news lately that most of you probably heard about. You've heard about so-called high-path bird flu, high-path avian flu. These are of influenza viruses that ultimately come from wild birds and adapt to domestic poultry like chickens and turkey or quail. They have an insertional mutation at the cleavage site between the 2 parts of the hemagglutinin that gets proteolytically cleaved. And that normally they're cleaved by a trypsin-like protease, but in the case here, as we saw with SARS-CoV-2, there's an insertional mutation where you get a run of arginines and lysines, basic amino acids that give you a furin cleavage site, which allows the viruses to be cleavage activated intracellularly by intracellular furin-like proteases that are present in almost all cells. So it allows these viruses to replicate systemically in the animals they infect, causing this to become a very pathogenic virus for chickens. So the highly pathogenicity aspect relates to how it relates to chickens, that it kills chickens, but does not necessarily mean that it's high pathogenicity in other animal hosts, just for clarity. It's not specifically derived as a human term. In the middle of COVID, when everyone was worried about COVID, H5N1 viruses that had been high-path viruses that had been circulating starting from an epidemic in 1996 in southern China. So for a 30-year period, the viruses have been circulating and that have occasionally spilled over into domestic poultry populations, causing devastating outbreaks, but also have spilled over into humans occasionally, but never transmitted human to human efficiently, that have been sort of smoldering along, that they exploded in the wild bird population and spread across most of the northern and now southern hemispheres all the way into Antarctica, and that you have a panzootic of high-path H5N1 viruses in wild birds. The interesting feature is that the major hosts of these, like ducks, different species of ducks, shed these viruses and yet are not generally sickened by them, and so spread lots of viruses. And there are other avian species that are not normally infected with influenza that are very susceptible, like raptors, owls, eagles, condors, and things, and they've had large die-offs of that. They've also caused huge outbreaks in wild mammals, which is something that's also really novel. And so this, over the last few years, there have been at least 50 species of wild and domestic mammals that have been infected with high-path flu viruses in the United States and in other places around the world. A devastating outbreak, for example, in sea lions in South America. Then we have this concerning issue over the last couple of years in which these high-path avian viruses have been detected and spreading in dairy cattle ranches. And so in the last couple of years, there have been over 1,000 dairy farms that have had outbreaks of H5N1 in 19 states, as shown on the map here, and that this has led to at least 71 people who are known who have been infected with H5 viruses in the last couple of years, 2 of whom have died, and about half of them have been exposed to dairy cows or milk, and a lesser number of them have been exposed directly from wild birds or domestic poultry. I want to say that in regard to adaptation to cows, that it doesn't look like the virus is really spreading efficiently cow to cow, that this is really kind of a human activity-based thing, that the virus, while it can cause some mild respiratory illness in cows, it generally causes a primary mastitis. And so in dairy farms, when these cows are milked with these milking machines, the virus is being transmitted cow to cow through the human activity, through milking and through sending cows and equipment and people who are exposed to these viruses from one dairy farm to another and across state lines. And so much of this spread is not due to the adaptation of the virus, but to human activity or the lack of appropriate human activity in causing this outbreak to extend so widely. But any time that an avian virus spends a lot of time in a mammal, you always worry about the possibility that a virus could emerge that has features that could bring it into further human infection. So maybe the paradigm should shift, and rather than the pig, we have to start worrying about the cow. So some unanswered questions. Will H5 adapt to transmit efficiently in cattle or in other mammals? Will it adapt to be transmissible in humans? Will it reassort with human seasonal influenza viruses? And in either of those cases, would it cause a pandemic? And if it caused a pandemic, what would its phenotype be? Would it be mild? Would it be severe? And the problem is that we don't have the answers to any of these questions, and unfortunately, we haven't done a great job with the infrastructure of determining where these viruses are through surveillance of wild animals, through domestic animals, and through people. We really don't know what the exposure rates have been, although wastewater deep sequencing suggests there's a large environmental load, or there has been over the last few years. Also, are there animal models that are more useful to accurately reflect the current situation that we're seeing in cattle, where they're having a generally mild recoverable disease with a primary viral mastitis and not a severe lethal infection? So to go from the unanswered questions that I just posed to unanswered questions from 200 years ago in a book about influenza that was written and published in German by Dr. Most, on the front page of his book, he says that he has a couple of questions that he wants to address, which is, what is influenza? Where does influenza come from? How did influenza behave in the past? In what ways can we predict the future occurrences, and how will the future outbreaks occur and behave? And through what means can its spread be halted? And while the topic of this overall session, as Dr. Arias has said, is demystifying medicine, I have to say that I'm still pretty mystified about the answers to these really fundamental questions. And these are still incredibly relevant questions. And it's very humbling that after 100 years of virology research on influenza viruses, we still, while we know tons of data, we still have a very hard time answering these questions much better than Dr. Most did 200 years ago. So I asked this question, can the next influenza pandemic be predicted? And the answer is no. I feel confident that there have been lots of influenza virus pandemics in the past. There's tons of influenza in hundreds of animal species all around the world. The likelihood of there being another pandemic is very high, but I can't predict, and no one else can predict when it will occur, what subtype it will be, where it will occur, or its level of pathogenicity. But so if we can't predict yet the future, can we learn something about the rules that influenza follows by looking at past pandemics? So we know about 4 pandemics in the past. So very briefly, just to talk about the 1918 flu, as Dr. Arya said, it caused a devastating outbreak with about 50 to 100 million deaths globally in a world population of around 1.8 billion, a little under 700,000 deaths in the United States. If you look at life expectancy in the United States, because of the 1918 flu, it caused life expectancy to drop by about a dozen years in 1918. It had an unusual age-adjusted mortality curve, such that there was a peak of mortality not just in newborns and the elderly, like you typically see with influenza, but a new peak, sort of a rough W shape, where there was a new peak of mortality in young adults, peaking at about age 28. Because of that, it had a disproportionate impact on the military, and in the U.S. military, of about 100,000 troop casualties for all causes in World War I, about 40% of those were due to acute influenza pneumonia deaths in young, healthy soldiers. So there were no influenza isolates made in 1918. The concept of viruses was sort of coming into being, and people were speculating that it might be a virus, but the viral isolation of influenza viruses did not occur until the early 1930s. So there was no way to actually address what the 1918 flu was, or to understand what the implications of that virus were. So as Wynn said, 30 years ago, we started a project to sequence it from archival material, autopsy tissue, starting with soldiers who died in the 1918 flu from the Armed Forces Institute of Pathology, and then expanding that to archival pathology material in Europe and other countries, as well as some exhumed frozen lung tissue in Alaska that was contributed by my colleague, Dr. Johan Holten. And this painstaking process was used over about a 10-year period to sequence the complete genome of the virus, and then it became possible with a multi-center collaboration to embark in a project in which the virus was reconstructed using reverse genetics, and infectious virus was made in high containment labs, initially at the CDC, and then after my moving here to NIAID in 2006 here at NIAID. So very briefly, it looks like the 1918 virus was an avian-like influenza virus that adapted to humans sometime before 1918, and that by many measures, its sequence, its phylogenetics, its GC content, the sequence of the proteins that it encodes, it is very avian-like, but whether it went through an intermediate host, we don't really know. That we now have over 20 influenza genomes, complete or partial, from 1918, and shows that while there's a minor variation here and there among the sequences, they're extremely uniform, so that this virus spread without much change in the early months, going from May 1918, the earliest isolate we have, up through February 1919. And while there's much that I could talk about, I'm only going to mention 2 particular lessons for 1918 that I would like you to take away with. One is that when you look at human autopsy tissues of people who died in the 1918 flu, and as well as when you look at the lung tissues of animals that are used in the laboratory to study pathogenesis of influenza viruses, like various inbred strains of mice or ferrets and non-human primates, one thing that was observed in all of these species is that the virus was very pathogenic. It didn't require any prior adaptation to be pathogenic, and induced an incredible pro-inflammatory infiltrate in which a huge number of neutrophils, as well as other white blood cells, flooded into the lung, which is very unique for a viral infection, and induced a very profound pro-inflammatory response with inflammatory mediator cell death responses and reactive oxygen species. And interesting, during the early period of COVID, when we put together a multi-center COVID autopsy study to look at the pathology of fatal cases of COVID in the early first year or so of the pandemic, we found that the early early COVID viruses up through Delta also in fatal cases induced a very similar, very pro-neutrophil, pro-inflammatory response. So the 1918 virus and the COVID viruses share some pathophysiologic features in terms of inducing this kind of inflammatory response. But the reason I say this whole thing is to make this point, is that the virus itself certainly causes damage and kills cells as it replicates down through especially the lower respiratory tract. But what you find is that a major contributor of the pathology is the inflammatory response derived from the host itself. So that if you infect mice with a lethal dose of the 1918 flu, and you take half of them and you treat them with a drug that blocks the formation of free radical oxygen species, but has no antiviral effect, the virus replicates to the exact same titer as the control, up to 10 to the 8th to 10 to the 9th plaque-forming units per mil in the lung. And yet there's no pathology, very little mild pathology, no free radical oxygen damage, and mice survive. And in the untreated animals, you get a rapid lethal pneumonia with significant free radical oxygen damage. So the key point here is that damage from the primary viral infection itself is survivable under the right circumstances, but the lung damage from the inflammatory response is actually a significant component of the pathogenesis and is actually lethal. The other second and last feature I want to mention that's important about the 1918 flu is that it really is the mother of all pandemics. So all the human influenza viruses of the last 108 years, whether they are pandemic viruses or the seasonal viruses in between, all the way up to this year's flu viruses, are all directly descended from the 1918 flu. So if you have tens of millions of mortalities during the pandemic itself in 1918 and 1919 from a single host switch event of an avian virus to humans, that virus has then been updated by mutation and reassortment with other avian and swine viruses over the last 100 years, but the core genes are still derived from 1918, so that the tens of millions of deaths of flu cases that have occurred over the last century are all also ultimately due to this single introduction of a virus. So we are in a pandemic era that is now over a century old, and we don't know how long it will last. But this shows you the importance of a single introduction event of such a pathogenic virus in 1918. So the lessons are that it likely derived from an avian virus, that it adapted to humans sometime before 1918 with or without going through an intermediate host, that its major virulence factor is that it induces this very strong pro-inflammatory response, and that you can actually treat animals with anti-inflammatory drugs that actually can save them, and that you don't necessarily need antivirals. And something I don't have time to talk about is also the critical importance of secondary bacterial pneumonias, but is also a major factor. So we are right now in flu season, and thinking about pandemic and seasonal influenza is really two sides of one coin, and we have to think about them at the same time. So if you look at seasonal influenza virus deaths over the last 10 years or so, what you find is that it's a very significant problem, that in some years you have over 50,000 deaths in the United States attributable to influenza pneumonias. In some years it's less than that, but it averages about 30,000 people in the United States per year, so it's a very significant public health problem. If you look at the rapidity with which these viruses evolve from year to year, you can see that in the blue line here, especially the H3 and 2 viruses acquire mutations that allow them to escape from pre-existing immunity, meaning that the vaccine strains have to be updated almost on a yearly basis to keep up with this rapid evolution. So we're constantly, in a sense, chasing behind the evolution of the virus. And the consequences of this is that the seasonal flu vaccines that we've been making for the last 60 years or so have suboptimal effectiveness, so that in some years you have up to 50 or 60% protective effectiveness, and other years it's in the teens. And so it's absolutely clear that we need to do better, but the question is how we can develop vaccines that would give us better breadth of and potency of efficacy. So you've probably seen in the news the term superflu. I don't know where it came from, but this is something that the news media has started to report, that we have a quote-unquote superflu that's spreading in the U.S. And what that is that we have a drift variant that has emerged. And so on the bottom graph, you see this, the orange component is the new subclade K of the H3 and 2 virus, which is really taking over now and is kind of probably causing other coexisting expecting H3N2 strains to go away, to go extinct. But the vaccine strain, when it was selected, because there's many months needed to go from vaccines to production to having vials to be distributed in for the flu vaccine in the fall. And so unfortunately, the virus that is now circulating has emerged basically after the strain selection event occurred. So we're going to have a big mismatch year. And so how it will impact in terms of morbidity and mortality is not clear and will only be clear in retrospect because we really can't predict yet. But it has the makings of being one of the worst seasonal flu years that we've seen, for example, like we saw in 2015. So the current flu vaccine strategies, of which there are many, from the live attenuated viruses, which had great promise but are used less because they have, probably being over attenuated, have not been exactly as effective as we would have liked them. Whole inactivated viruses, or then you can split the virus so that you have bits of the membrane with both the hemagglutinin and neuraminidase, or so-called subunit virus, where you actually just release and cleave off the major surface proteins, hemagglutinin and neuraminidase, and this is what's used most, and more recent technology to look at recombinant production of the proteins of the hemagglutinin. And then there are next generation platforms, including mRNA and viral-like particles, peptide-based, viral vectored, other things. But in general, for seasonal flu and for past pandemic flu, the goal has been generally to think of the hemagglutinin protein as the major dominant antigen, and that the goal is to have a vaccine that generates an antibody response measurable in the serum against the hemagglutinin protein. And the problem is that you can already see is that we're chasing behind the evolution of the virus, not getting ahead of it. So if you think about the problem of vaccines to respiratory viruses, I think if you look at a table here of human respiratory viruses, you can divide them into two groups. There's those that replicate systemically, like measles, mumps, rubella. These are viruses with very long incubation periods of around 2 weeks and have a marked viremia, where the virus is circulating systemically in the blood and throughout the body. And then there's a group of respiratory mucosal-only replication viruses, the endemic coronaviruses, influenza viruses, paraflu, RSV, and for the most part, SARS-CoV-2. And they differ in that they have very short incubation period in just a couple of days, and they do not have a productive viremia. And so if you look at that, you find that those that replicate systemically, natural infection and vaccination create herd immunity and lifelong immunity. Whereas those that replicate only in the respiratory tract, herd immunity is highly elusive and vaccine control is problematic. And so I think that what that's saying is that for viruses that are in the respiratory tract, especially the upper respiratory tract, I think that what we're fighting is that there is a sense of tolerance that develops in the respiratory tract. Since you're breathing in foreign proteins all the time, you can't probably overreact with an inflammatory response to viruses, so it's probably physiologically more important to not have such a profound immune response. And that means that it's going to be very difficult to come up with strategies that would give protective immunity for these sorts of viruses. And we might have to think about mucosal levels of protection instead of just a peripheral intramuscular vaccination. I want to just mention Alexander Langmuir, who was the chief epidemiologist of the CDC from the late 40s up until 1970. And in an article published more than 60 years ago now, he reviews the evidence from seasonal flu control with the vaccination strategy, and he says that there's little evidence that recent vaccines have prevented clinical illness or effects on mortality, no scientifically valid basis for predicting antigenic shifts in the virus, and that sounder bases are needed for an influenza control program. This does not make either Dr. Langmuir or myself in any way anti-vaccine. What it suggests is that the current vaccine strategies are, while useful and efficacious, are suboptimal for what we really want to control. And so we need to look at ways to make influenza vaccines more broadly protective. So I'm going to spend the last couple of minutes just talking about an example vaccine that I've been working on here. There are many other very interesting and promising universal influenza vaccines that are being developed in other laboratories, but I'm just going to briefly mention an idea here and then pull it together. So one of the things we think that's very important is that we should be studying the immunology of influenza in the hosts that we care about the most, which are people, rather than what we learn in animal models. So to do this, we have been doing a series of studies for the last 15 years, looking at volunteer influenza challenge studies where young, carefully screened, very healthy young volunteers are recruited as participants in studies in which they are inoculated with wild-type seasonal influenza viruses, H1s, H3s, and that this is used as a basis for then doing early phase 2 studies to look at vaccine or therapeutic efficacy and to assess breadth of protection. So I think that it's crucial to take us from the laboratory into the clinic where possible to study influenza pathogenesis and immune and inflammatory responses in humans directly, and that understanding these processes are critically important for the development of prognostic and diagnostic markers, therapies, and next generation vaccines. So as an example, when you are infected with influenza very rapidly, by 24 hours after infection, you have a lot of virus. By 48 hours, you have a very large amount of virus in your nose and throat, and yet you don't have any symptoms. And so for the first couple of days, you're shedding virus basically asymptomatically. And so this is a time where you could think about doing biomarker discovery for either diagnostic or prognostic efficacy, because it's very difficult to determine if someone presents with an acute influenza infection whether they're going to have a mild and easily recoverable illness or a long-term illness that would require hospitalization. So looking at peripheral blood mononuclear cell transcriptome analyses, we can find that in the 24 to 40 hour pre-symptomatic time point, you can make very good predictions as to who is going to have the most severe influenza disease, use this sort of as a prognostic marker. But we think that since the infection is occurring in the nose and throat and the upper respiratory tract, that we wanted to look more specifically at what was happening directly where the viral infection is occurring. So in that case, with our collaborators here, we did another example challenge study with kind of a systems biology approach where we took a group of people, and half of volunteers in this participant study, half of them were vaccinated with the seasonal flu vaccine from that year, the 2018. Half of them were unvaccinated. They were then all challenged with the matched H1N1 virus that would be in the vaccine. These are all young, healthy people. And then we did extensive sampling of nasal mucosal samplings, looking at antibodies, looking at cytokines. We're looking at gene expression. The same thing with peripheral blood, looking at T and B cell repertoire, looking at T cell function, looking at inflammation and immune markers, looking at antibodies. And so what we find is that you can, of course, with nasal gene expression, see very good correlations, again, very early on, that correlate with daily symptom score and viral load, and you can have both very good prognostic markers looking directly where the infection is occurring. And that we are able to put together this sort of model of the components of both the mucosal respiratory as well as the systemic immune responses, and how both of them play critical roles, but different roles, in an acute influenza infection and recovery or neutralization of infection. So we can divide people who don't shed virus, who have neutralizing antibodies at a titer high enough to protect them, and those that shed for a very small amount of time, only one day, don't have high enough titers of neutralizing antibody but have memory T cell responses that allow them to recover quickly, and those that have multi-day shedding lack both adequate B cell and T cell responses. I would like to point out on the right, though, however, that in these very healthy young people, average age about 28 or so, that 62% of those that were vaccinated shed virus, which was the matched virus of the vaccine. Again, showing how this vaccine is really suboptimal in terms of limiting viral replication. And so doing these kinds of studies in humans, I think, is going to really help us think about the next generation of flu vaccines. So in the last couple of minutes, let me talk about universal or broadly protective vaccines. The word universal flu vaccine can mean different things to different people. It could mean breadth of protection in terms of the different viruses, all the different kinds of human and animal viruses that you could protect against. It could mean what is the breadth of protection in terms of the different kinds of people, the elderly or children or those with comorbidities versus young, healthy adults. Are different populations and might need different vaccine strategies. In terms of breadth of protection, in terms of the mechanism of the protection offered, I think enhancing mucosal immunity would be very useful. Having a variety of responses and including antibodies to neuraminidase and other proteins, enhancing T cell responses, especially mucosal-based T cell responses, I think would be very useful. So to do this, we've adopted a strategy that seems to work very well in preclinical data, and it's in early clinical development, and we'll see in the next year or so how well this does in people and how this translates to other potential universal vaccines. But this is a whole inactivated virus that takes 4 low-path avian viruses of 4 different hemagglutinin subtypes and 4 different neuraminidase subtypes, and chemically inactivates them with a chemical that induces mutations in the RNA but doesn't really change the structure of the protein, so that you get good presentation. The virus still looks like a virus. It's taken up. It binds to the receptor. The neuraminidase enzyme is still active. The virus just can't replicate because its genome has too many mutations. And what you find is that this cocktail with 4 different hemagglutinin subtypes and 4 different neuraminidase subtypes in mice and ferrets induces good antibody responses to the hemagglutinin antibodies, neuraminidase, M2 antibodies, and induces good T-cell responses. And used either intramuscularly or intranasally, offers extremely broad protection using challenges of up to 50 times the lethal dose of challenge viruses of all subtypes, including subtypes that are not in the vaccine at all. So completely novel viruses that you wouldn't expect to see protection, but we see basically 100% protection from almost all these challenge viruses. As an example here in ferrets, you see that here's an example of challenge with 4 different viruses, 2 of them, the H3 and 2, the H10 and 7, the H2 and 7 and the H10 and 7 are completely heterosubtypic in these ferrets from the vaccine. And yet in all cases, you see that viral replication is knocked down really significantly, that there's really very little host inflammatory response induced either intramuscular or intranasal vaccination. And most importantly, there's full survival and there's no viral pneumonia developing. We can see good correlative protection in terms of serum and mucosal IgG and IgA. We see increases in lung cytotoxic CD8 and then CD4 cells. We see increases in NK cells, and we see increases in lung resident memory T cells. We know that heterosubtypic protection, that is protection against completely novel viruses that are not present that have antigens that are not present in the vaccine, requires a T-cell help, that its antibody alone does not save animals from that, so passive transfer of serum will not work. So it shows that it requires both B-cell and T-cell activity to achieve the kind of protection we're seeing. This recent H5N1 viruses are unbelievably pathogenic in ferrets and mice. These viruses cause rapid lethal encephalitis in mice and kill animals in just a few days. But the vaccine has an H5 component, but it's very different than the H5 viruses that are currently circulating in birds and cattle and the occasional human now. And so what we find is that we get near 100% protection with this vaccine in H5 lethal challenge, 10x lethal challenge in mice. In this one study here I'm showing here, 14 of 15 mice survived, and the one mouse that did not survive did not seroconvert, so the vaccine was probably a miss in this case. And that the H5 sequence identity between what's in the vaccine and what's in the challenge virus is almost as different between 2 HA subtypes, between, say, H5 and H2. So in the human phase 1, what we see is that there were no significant safety issues, had an excellent safety profile, both by IM and intranasal routes, that we saw the IM generate decent antibody responses in the serum against the 4 HAs and the 4 neuraminidase, and the same in the nasal. We saw antibodies against IgG and IgA against the HAs and the NAs for the intranasal. So we are going to start a phase 2 efficacy study where vaccination followed by challenge with human H1N1 viruses and other subtypes to follow later in 2026. So I end by saying that ideally what we could do is in a multi-center way, we could evaluate example influenza vaccines like the one I've just described and other candidate vaccines in an apples-to-apples way by doing these early phase 2 challenge studies with humans in which you can do a kind of a systems biology approach and you can evaluate vaccines, you can develop look at all the correlates of immunity if we sample all in the same way, so that if different vaccines are evaluated in the same sort of clinical trial where all the same kind of mucosal and respiratory and systemic immune sampling occurs, host factors, and yet a toolkit of antigenically variant challenge viruses that we could use to evaluate breadth of protective efficacy, including even low-path avian influenza viruses. We have been able to evaluate in the clinical center here in the isolation ward a human challenge model with a low-path H10 virus, which causes very few symptoms, but does replicate for a couple of days. So there is something that could be measurable in terms of sort of measuring its ability to be kind of a pre-pandemic virus. So I will end with that, with the final thought of to keep calm and get your flu shot, or maybe in the future a spray up your nose. And I will end by thanking all the people in labs and in Matt Memley's group who have worked on the aspects of the projects I've described, as well as funding from the NIH, HHS Central, BARDA, DARPA, and the Gates Foundation. So thank you.
Oh, thank you very, very much, Jeff. I think, Arturo, are you ready?
I am as ready as will be. I'm going to try one more time to see if I can share. Yes. It worked.
Okay. Whew.
Can you see my slides?
Yes, I can.
All right. So for the audience, we spent quite a bit of time trying to share and nothing worked. And then something magical happened while Dr. Daudenberg was speaking, and it works. So I'm going to be talking about a completely different topic, and that is with the climate change bringing new infectious diseases. I'm going to touch a bit on the idea of fungal pandemics. We haven't had one yet. We hopefully won't have one, but certainly they could happen, and I'll tell you how that may be. So I have no disclosures, and I'd like to begin with the statement that global warming is a fact. That is the consensus of the scientific establishment, and those of you who are experiencing warmer winters and where rivers don't freeze as they used to, or lakes freeze as they used to, are witnessing global change in your life. We are the first humans to witness climate change in our life, because in the past it always occurred very slowly, such that generations wouldn't even notice it. The important thing is that changes in temperature will change all aspects of metabolism. And in fact, the reactions in a cell are given by that famous formula. The delta gives free energy, and temperature enters into the equation. You change 1 degree, you change all aspects of cellular function. It means that all organisms are going to, whether they are macroscopic or microscopic, are going to have to adapt or die, and that includes us. And global warming will have great effects on the microbial world. So I want to take you back 250 million years. And I hope this doesn't happen, but there is some way to think about what may be happening in the context of what happened then. 255 million years ago, the biosphere experienced the greatest extinction event, which is known as the Permian. 90% of our species died. And what happened was that a coal fire, yes, there was coal 250 million years ago, began in what is now Siberia. And it burned for 80,000 years. And during that time, it dumped enormous amounts of carbon dioxide into the atmosphere. It changed the climate. The world changed, and most species, even though they had 80,000 years to adapt, could not do it. We are doing the same thing in a couple of centuries. Now, even though the biosphere was wrecked, there was one group of organisms that did well, and those were the fungi. The fungi do well whenever there are major calamities. This is a mountain in South Africa, and you would see there on the left an area which is called the fungal spike. This is about 1 meter of fungal spores that you can still find from 250 million years ago. And all life that survived had to live in a fungal world for a while. So that brings me to the concept of disaster microbiology. When you hear about tornadoes, when you hear about tsunamis, we think about the destruction of the large structures. But it happens that all the microbes are also altered whenever there is a disaster. And global warming is, in fact, a global disaster. It will affect everything. But this is a concept that is important because we don't often think that the disturbance in the microbiology will have consequences for us. And yet, when you look at disaster after disaster, you find new diseases, you find new problems that are often associated with the change in the unseen world of microbiology. So higher temperatures will affect both the host and the microbe. The microbes will be stressed. They're going to have to adapt to higher temperatures. And one of the fascinating things is that they often will acquire drug resistance. How is drug resistance tied up to temperature? Well, temperature is a stress. Drug resistance is stress. When organisms develop the capacity to resist one stress, they can acquire to resist other stresses. And hosts we know very little about what happens to hosts. In fact, very little science has been done to understand what's going to happen to humans if you change their average temperature in which they live. But the initial evidence is not good. It increases stress. This increased stress reduces immunity, and you have more inflammation. And as we are learning, inflammation is something that destroys organs. Jeff just mentioned how the problem in flu is the inflammatory response. Well, systemic inflammation is going to have a lot of health consequences. And now I ask you, where do pathogenic microbes come from? Well, Jeff just told you about flu and told you about COVID and told you about viruses. All those viruses come from other hosts. Other pathogens like bacteria and parasites come from other hosts, but some pathogens come directly from the environment. And certainly viruses can't come from the environment very easily because they got to come from other hosts. Some bacteria like Legionella, you will get it from the environment. Some parasites, free living, like Giardia, but almost all the fungi that cause disease come from the environment. So you acquire them directly from the environment without intermediate hosts. The fungal kingdom is the largest kingdom. This is old terminology, but it works. It's got over 6 million species, and the majority of them have not been described. They are the major pathogens of plants, insects, invertebrates, and ectothermic vertebrates. Those are cold-blooded vertebrates. They're currently destroying entire ecosystem. The bats in North America, the amphibians throughout the entire world, salamanders in Europe, snakes in North America. The point that I'm getting at is fungi can cause pandemics. They don't affect us very much. And the question is, why? So mammals turn out to be remarkably resistant to fungal diseases, and we're going to explore that a little bit. Not only are mammals highly resistant, but we have relatively few fungal species that cause disease. From the host-associated, these are the ones, Candida, we tend to get from whoever took care of us when we were a child. Dermatophytes are in our skin. Those cause athlete's foot. They live there. And then there are organisms like pneumocystis that we don't understand the epidemiology very well. Those we tend to get from other hosts. But the great majority we get directly from the environment. And those are organisms like Histoplasma, Aspergillus, Cryptococcus, Coccidioides in the southwest, and Blastomyces. So it turns out the mammalian temperatures protect us against fungi. This is work that we did more than a decade ago. But on the left, what you're seeing is when we looked at 4,000 species of fungi in a collection and asked what temperatures can they tolerate. Most fungal species do very well up to about 30 degrees, but they rapidly dropped after 30 degrees their ability to be viable. So for every degree above 30 that you go up in temperature, you can exclude 6% of fungal species. That's telling you the amount of protection that you're getting by walking around the world at 37 degrees. Now, my friend Aviv Bergman took that data, and he took a well-known equation for calculating the cost of being hot. And he asked, what is the temperature by which you can keep out most fungal species and don't have to eat all the time? And he found that that temperature was 37 degrees. Now, that suggests to me that something in the past selected for this temperature. After all, the mammalian lifestyle is very expensive. So the mammalian defenses against fungi, you have this enormous temperature barrier that keeps out most of the kingdom. And then whatever gets through can be handled pretty well by humoral and cellular immunity, provided that the host is not immunocompromised. So today, most invasive fungal diseases occur in hosts with impaired immunity. And if you look at the majority of the diseases that we're familiar with, nail fungus, athlete's foot, dandruff, which is a fungal disease, or babies getting diaper rash, these tend to occur in the cooler parts of the body. So the cooler parts are more susceptible to fungal disease. Now, we have John Perfect at Duke and others have carried us a remarkable series of experiments to show the importance of temperature and where temperature and immunity come in. So bunnies are resistant to a fungus, Cryptococcus neoformans. You cannot give them systemic infection. You can, however, give them a skin infection or a testicular infection because there's a cooler. But if you give steroids, temperature is not enough, and you get a dead rabbit. So the emerging pattern is this remarkable resistance. Why most of us in this lecture don't worry about dying of fungal diseases is coming from this combination of immunity and temperature. An example of this comes from the calamity that has befallen the bass in North America. Bass in North America were doing well till 2006. And then a fungus was introduced from Europe that was first seen in a cave in New York State. This fungus grows on the bats when they hibernate. When they hibernate, their temperature drops to 12 degrees. It basically grows on them, and it's called white nose. You've seen the hypha. You've seen the fungus growing on the nose, and it kills them. If you take these bats into the laboratory and you feed them and raise their temperature to 37, they can clear the infection. Temperature is very powerful against the fungi. Another example is the calamity that has befallen the amphibians, which are being decimated by a chytrid fungus. If you create saunas, that is warmer areas where they can take refuge, they will go into, for example, a cinder block that is heated. The frogs will climb on it, and just raising their temperature allows them to cure the disease. So one of the questions that always fascinated me is, why mammals? Why are they so resistant to fungi, and why are the large animals? When you drive around, what do you see? You see humans, and any animals that you see tend to be mammals or birds, and they're both hot. Their temperature is hot. Now, the mammalian lifestyle is extremely expensive and only took off after what is known as the KT event. That's the asteroid that hit the Earth and wiped out the dinosaurs 66 million years ago. So on the left, you see energy requirements, and that's the metabolic rate by mass. So it's normalized by mass. We are up there with the elephants. And notice that a frog needs about 1,000 times less energy to get through the day than you do. A fly needs 10,000 times less energy. So I basically been fascinated. How does nature select for a lifestyle that is so expensive? I already had 3 meals, and I'm going to have another one in about an hour and a half. I'm probably at midnight snack. It's an enormous amount of consumption of food just to maintain my temperature. And these great mammalian radiation takes off after the KT event, and you see that on the right. You see the rise of the birds and the rise of the mammals, everything from whales to humans. So what happened at that cataclysm? Well, it's pretty well established that a rock from outer space hit the Earth. And what you see here is that you had a world of conifers and flowering plants. And then something happens at the KT boundary. And what you see, the iridium goes up because that's a heavy metal that's brought in by the meteorite and spread all over the world. And then the world kind of dies, and you only have fungi. And eventually it begins to recover, and you have the ferns, and then it took a long time before the forest returned. But we know that we had two types of survivors. We know that we had small mammals survive, and we had reptiles. And about 20 years ago, I proposed this idea that the reason that mammals take over is because this fungal bloom kept down the cold-blooded reptiles. They didn't kill every last one of them, but what happened is, after the calamity, the small mammals had an advantage, especially with all those fungal spores. And they had larger populations, and when the skies cleared and the earth recovered, there were just a lot more mammals. So you end up with a great mammalian radiation. So this was written 21 years ago already, and I update it every now and then. It has a lot of explanatory power. It's internally consistent. By the way, I haven't had any pushback yet. It could be that nobody really cares. But to me, it provides an internally consistent idea. It provides a selective mechanism for an energetically costly lifestyle. It explains why most mammalian temperatures are in the high 30s. It explains the tremendous resistance of mammals to fungal disease. It argues that our temperature is a major host defense mechanism, and it explains why doctors always give you Tylenol when you have a fever. The reason is that most of the organisms that cause disease in us today are already temperature adapted. Jeff's influenza, SARS-CoV-2 are temperature adapted. So we can't protect against that. But go on a hike, fall down, get a gash in your leg, and get dirt into it. Those 2 degrees could mean a tremendous amount of protection against environmental organisms. But then we have a problem. And the problem is that as the world gets warmer, the fungi are adapting to temperature. Today, most fungal diseases are near the equator. Why would that be? Well, statistically, the probability of getting a fungus that is adapted to temperature and has all the virulence factors to cause disease is more likely the warmer the climate is. Fungi, however, can be rapidly evolved in the laboratory for thermotolerance. What you see on the right here is a report from a company in which they were trying to get a fungus to be a better killer of insects. And the problem is that insects know when they get infected and they find a hot rock and they treat themselves. So they were able to adapt this fungus as a pest control to grow at much higher temperatures, 36 degrees, over a matter of months. Fungi have tremendous capacity to adapt to higher temperatures. We have some evidence that the fungi are already adapting. And this comes from a collection in which they record the temperature of all the isolates when they deposit it. So this is noisy and it's messy, and there are multiple curves there. The top curves are the Ascomyces that serve as a control. They're already temperature adapted, and they're really swinging, but they're not going anywhere. But the Basidiomyces, there is a definite trend upward with time. They appear to have been, we may be arguing about climate change, but the fungi have been adapted since the late 1980s. Now let's think of cities as heat islands. Now your typical city, most of us live in cities, is 4 to 5 degrees centigrade warmer than the environment. And there has been some evidence that if you look at a fungal species from a city and the same species from the environment, that the ones that are in the cities are already more temperature adapted. So we wanted to see whether this could happen in Baltimore. And David Smith, who's a postdoc in my lab, set out to look at this. Now, that's a picture of the Inner Harbor. I think it's a very nice city. And on the right is downtown city Baltimore, and you can see the number of hot days and the number of really hot days. And they are certainly going up with time. So what he did is he went to a cooler neighborhood, which is Guilford, and he went to a warmer neighborhood. By the way, that line, those of you who know Baltimore, that red line is Fayette Street. Fayette Street near Hopkins, where we work, is the hottest part of Baltimore. And he began to collect fungi from the environment. And what he found was that the fungi from the cooler neighborhood, the warmer neighborhood, was shedding their pigment. They were getting rid of their melanin. Why? Because melanin absorbs heat. And so they were already turning albino. And this is happening within 2 miles of each other. But the more concerning thing is that he found that an organism that is a opportunistic fungus in fact was more thermotolerant from the hot neighborhood than from the cooler neighborhood. So this has just been published in the ISME, and it tells you that within a city, neighborhoods that differ in temperature, you're already seeing fungal adaptation. So about 15 years ago, I warned that this could happen. That in other words, that the fungi, there are many fungi out there that cause disease in plants, insects, invertebrates, other invertebrates, and that are fully loaded with virulence factors, but they cannot touch you because they don't have the capacity to grow at 37 degrees. But if they adapted, then we will see new fungal diseases. So what I'm about to tell you is I think this already happened, and it's already happened with a new fungal section that is spreading throughout the globe known as Candida auris. So here are some facts about Candida auris. It was first isolated from the ear canal of a patient in 2007. And auris means ear in Latin, and that's where it got its name. So it's a new organism. People said, well, maybe we missed it. So they all went into their collections, and it wasn't there. An occasional isolate was found in Japan, in South Korea, but this was a new organism to medicine. And then comes the big mystery: simultaneous emergence in 3 continents. In South Africa and India in 2009, in Kenya and China, and in 2012 in Venezuela. And the isolates are not related. This is not because somebody took a plane and flew from one place to the other. These are new emergencies that are coming out of nowhere in the environment. And the question is, how do you get a new pathogenic fungi emerge? How did it emerge independently in 3 continents? Well, there are 3 theories out there. First one is it was there all the time, but people missed it. Problem, no. This has been looked at. Retrospective analysis shows that it was not there. The organism is new to medicine. It was a consequence of the overuse of azoles in medicine and agriculture. Problem, this doesn't follow epidemiologically. The azoles are not necessarily where the sites this is coming. And it's also unclear how antibiotic selection will lead to a new virulent species. And we have proposed that it's the result of climate warming, which selected for variants that could grow at human temperatures. And the problem here is that this is conjecture, and all evidence is going to be circumstantial. And any sort of proof is not, you're not going to be able to prove it with a smoking gun. But let's go and let's see how far we can get. Well, one thing you could do is you could look at its nearest relatives, and you could say, are they more temperature adapted? And the answer is no. It appears that this organism has, this species has the ability to rapidly adapt. It's not really, as I said, not known to medicine. It's resistant to 2 out of the 3 major classes of antifungal drugs. And the isolates are not genetically relevant. Now, since 2013, there have been 3 other emergencies, one in Iran, one in Russia, and last year in Singapore. And again, they're not related. So this thing is basically emerging from the environment without really explanation. Now, a hint that this may be global warming comes from the work of Anura Chaudhry, who recovered it from the environment. So she found it in the Andaman Islands in 2 places, in a beach that was frequented by people and in a marsh that was very remote. The organism from the marsh has trouble growing at 37 degrees. What this suggests is that some of the wild organisms are not there yet, but they are, in fact, adapting. So I think Candida auris may be the first example of a new fungal disease from climate change. And now we have another problem. We are colder than our grandparents and great-grandparents. This is a fascinating paper that was published a few years ago, and it shows that our temperatures are decreasing with time. How can that be? Well, it turns out that, I guess the answer is we don't know, but the most likely explanation is that our grandparents and great-grandparents lived in a different world in which there were a lot more infections and there was a lot more inflammation, and inflammation drives up temperature. So another problem is that global warming, and every time you increase the temperature, you increase mutagenesis. In Cryptococcus neoformans, if you increase the temperature to 37 degrees from 30, all the transposons begin to jump. And most of these jumps are really deleterious. But an occasional one increases drug resistance and virulence. So we're not only having adaptation, but we're having genetic changes that can give us new virulence phenotypes. So I'm almost at the end, and I can't resist but talk about the big hit that put the fungi in many people's mind, and that was the series The Last of Us. So this was a series that played a couple of years ago in which a fungus adapted to climate warming by turning people into zombies. And it was inspired by the fact that there is a fungus that can turn ants into zombies. It's a cordyceps. It's a remarkable fungus. It basically goes into them, replaces their nervous system with hyphae, makes them climb up a tree where they die and they sporulate, and all the spores fall down to cause more infection. So when this came out, I had 15 minutes of fame. Many reporters were calling me and saying, well, were you involved with this? I said, I had no idea. I haven't even seen the series. But they always ask me the question, doctor, could this really happen? And I said, well, it's improbable, but not impossible. They said, what do you mean? I said, improbable. I don't know of any fungus that can turn a human into a zombie, but not impossible, because we know that a fungus can turn an ant into a zombie, and we know that fungi make things like LSD, and they make hallucinogens. How do you know that in the millions of species out there, there isn't some species that can make some compound that can turn you into a zombie? I don't know. So that was always the answer I gave, and I don't think I satisfied them, but at least I thought it was scientifically, I wouldn't say accurate, I would say plausible. So my last slide, I want to close by saying that mammals make no sense without the fungi. To me, when you look around and you see birds and mammals, something had to select for a lifestyle that is so expensive that if we were reptilian, we only had to eat once a week. Fungal selection after the KT event could explain the mammalian resistance to fungi. We are resistant to them because they selected for us. However, the thermal barrier that protects mammals is narrowing. The world is getting warmer, and the fungi are adapting to higher temperatures, and the humans are getting colder. So I think that this is heading into trouble. Humans are getting colder, fungi are adapting. We already have Candida auris as an example, and I think that there is trouble ahead. When you think about global warming, don't think about the 1 degree or the 1.5 degree average. Think about the number of very hot days. Next summer, when the temperature goes over 100 degrees, think about that as a selection event. All the organisms in the soil, in the sidewalk, are going to have to make it to that temperature. And the temperature on the sidewalk will go much higher, 130, 140 degrees. So how do we prepare for this potential nightmare? Well, I think that if you listen to Jeff talk, think about how much we know about influenza and how little we can really predict. I don't think that we can predict what's going to be the next one, even here. In fact, the state of the science, the state of knowledge of the fungal world is in fact much less sophisticated because it's so large relative to what we know about viruses. But I do think that it would pay to sample the natural world, to know what's out there, because we currently don't even know what's in our backyard. And I think it will pay to do more studies as a function of temperature. Microbiologists tend to grow their organism at the preferred temperature. Immunologists study everything at 37 degrees. even though my hand is about 7 degrees colder. Is the immunology of a paper cut the same as the immunology of a 37 degree? I don't know, but I think we need to study and learn more about that. And I think it may be possible to develop antimicrobial drugs that target temperature adaptation. Think about it. A fungus goes into your lung. It's used to living in the environment. It now has to adapt to much higher temperature. Turns on all kinds of programs to survive. If we can target them, you can basically make them not temperature adaptable, and that will give you a whole set of drugs. And with that, I thank you for your attention, and I am so glad that I was able to present to you, at least without using the PDF that we made. So thank you, and I will stop sharing so that I.
Well, thank you both very much for, my gosh, extremely exciting, provocative, and most informative presentations. It's a great way to start off demystifying medicine with such major challenges. So we have a bunch of questions, and I'd like to circulate some of them. One of them, a colleague asked, what does efficacy actually mean when it's reported about, I suppose, a vaccine or a drug or anything? Does efficacy mean prevent infection or decrease symptoms or prevent death, or what's the definition of efficacy? I don't know. Jeff, you want to answer that one?
It's a good question, and the answer is all of those things could be considered. It depends what you're actually measuring. So you can see, for example, that seasonal flu vaccines still allow people to shed virus, but the efficacy that's usually measured is reduction in clinical illness. So even though these people shed virus, they may have less symptoms or they're much less likely to be hospitalized. So, it really depends on what you're trying to measure. In my view, the most important feature, say for a respiratory viral vaccine, is the most important measure of efficacy would be reduction of serious illness. That if you could reduce the number of people who have to be hospitalized and reduce the number of people who would otherwise die and are left with just a recoverable infection, I think that would be a big positive. But again, it can be how it's being defined. But in general, when they talk about the vaccine efficacy, in this case, it's looking at a clinical infection.
So there's a question about Arturo, the presentation that you make regarding the profound effect of increasing temperature on adaptation and resistance of fungi. Does that apply to other living forms, viruses? That kind of adaptation brought about by a rising temperature? Do we have, what's the state of knowledge of the effect of either rising temperature over a long period of time, or just as you point out, the effect of an hour? What do we know about that in terms of viruses and bacteria?
Very little, very little. I mean, I think virologists have looked at temperature in terms of attenuating viruses, and Jeff can talk about that. But I would say to you that a grasshopper that has a viral infection has to make it through a hot day that virus has to adapt to. So I think that you will see adaptation in all microbial forms of life, whether it's parasites, bacteria, viruses, and all that. And we know very little. What I have been the most surprised at has been that the little work that has been done shows that for a complex eukaryotic organism, like a fungus, that the adaptation that is needed to go up a few degrees is only a few mutations. We are not talking about wholesale changes in the genome. We're talking about some strategic changes in some mutations and regulating of heat shock proteins and things like that. And you can dial up survival temperature. So this is not going to be hard to do.
So, Jeff, what do you think about that? What are your thoughts about this question that Arturo just responded to?
I think it's incredibly fascinating, and his whole lecture was fascinating to me. Had I known that it was his topic, I could have altered my lecture a little bit. There's some fantastic work that Stephanie Williams in my laboratory, who just finished a PhD at the NIH Oxford-Cambridge program, has been working specifically on temperature adaptation of viruses as part of avian influenza viruses adapting to mammals. Birds generally have higher core temperatures than mammals, and especially that these viruses in birds replicate in the GI tract. So at the core temperature of these birds, often as high as 41, 42 degrees or higher. Where in mammals, what you want is, in a sense, if you could give intent to an influenza virus, it has to be spread person to person. You want it mostly in the upper respiratory tract so you can cough, sneeze, and breathe it out. And so the viruses have to cold adapt to adapt to mammals, and that has been part of this process, as Arturo said, of cold adapting viruses to make attenuated viruses for vaccines. But we've been studying the fact that it's interesting that the 1918 virus is very avian-like, and yet it works in mammals. But it's interesting that its RNA polymerase gene is very avian-like. And interestingly, both the 1957 and 1968 pandemics also acquired novel avian-derived polymerase genes, PB1 genes, from a bird source as part of that pandemic. And so was that random or just an interesting selection factor? And so what she and her colleagues have found in some very elegant work is that these avian viruses, their polymerases, function much more efficiently at the bird temperature at 40, 41 degrees, and do not function well at the upper respiratory human tract temperatures of like 35, 34 degrees. And so one of the reasons that pandemic viruses that get genes from avian viruses might be more pathogenic is that they replicate better in the lower respiratory tract. And in fact, if you mount a fever response, which is a classic antiviral response, it may paradoxically cause enhanced viral replication of an avian virus in humans. And so that's been one of the really interesting features of looking at the 1918 virus and as an avian virus adapts to humans. But I just wanted to, if I can, just quickly ask Arturo what, in terms of we've been thinking about how climate change is going to affect the migration of birds and animals and these other things. But your point about how the viruses are going to be adapting too, it's clear in our case that flu viruses can readily adapt over what our large temperature ranges, say, 8 or 10 degrees C range, from 43 to 35 degrees. And so do you see that you would likely, as you just said, think that there will be more rapid evolution of viruses as well with increased temperature?
Yes, Jeff, I do, because it basically will dial up everything. And as you know, this is, I was aware of your work on this, which I think is really important, and I love that idea that a fever could make you worse in terms of replication. But I do think that you probably will also see increased mutagenesis if you were to do your experiments at higher temperature. I mean, I am sure your lab works primarily at one temperature, just like most lab tends to work. The fungal grows at 30 degrees. All immunology is done at 37. But God, it may very well be that fevers and passages through a warmer animal also is giving you an increasing mutagenesis.
No, we agree, and we are touching on that a little bit. And it's a really important question. Thank you.
So, Arturo, an attendee asks whether you have seen an evolutionary trade-off in the fungi growing more efficiently at higher temperatures. Is there some form of evolutionary trade-off for that gain capacity? And is there an impact on fitness? And could that be a target for future antifungals?
So the second part, the last part is easy. I do believe, so the fungi get totally stressed once you take them out of normal temperature. In other work that I didn't present, I can tell you the fungal world is hypothermic. That is, mushrooms in the forest at a lower temperature. They maintain a cooler environment. So when a fungus gets into a mammal, it's very stressful for them. And the fact is that the majority can't handle it, and they just don't survive. But the question of a trade-off, I think in one of the things we learn is there is always a trade-off. If you adapt to higher temperatures, probably something else will go. People often ask me a variation of that question. I said, "Oh, gee, if the fungi are so adaptable, how come they haven't adapted to eat mammals yet?" And I said to them, "Well, where is the evidence that virulence is good for a microbe?" I would argue that it isn't. I think that the ability, once a microbe becomes dependent on a host, then its evolutionary life is dependent on it. If birds and mammals are wiped out, influenza virus is wiped out. It's so acquiring virulence and acquiring dependence on a host is not necessarily a good long-term strategy if you're a microbe.
We have a question for Jeff here. What countries today do you think are at the greatest risk for an emergence and spread of the HPA1 strain? And what can NIAID do to address this global threat at its source? Do you want to comment about that?
Sure. No, good questions. So one of the problems is that these high-path viruses, this H5 virus as an example, these are viruses that really only emerge in poultry. It's an adaptation, this mutation that allows the virus to replicate systemically, seems to somehow only happen in poultry. But something odd happened underneath COVID, which was that there was a back adaptation of an H5 high-path virus from chicken to wild bird species. And the virus has reassorted itself multiple times with different gene segments from wild bird viruses and has moved around and is now spread through wild bird populations in all continents. And so these viruses are really panzootic, that is, to use a sort of pandemic, but for animals, a panzootic of H5. And so it's absolutely impossible to imagine eradicating it. So that's the first part of that. What we can do is certainly, it's critically important to do surveillance to know where these viruses are. So that is not always in NIAID's portfolio. It involves other state and government agencies, like the USDA example. But that's really important. And like I said, I think that it would be impossible to say which country is at most risk since these viruses are everywhere. The United States has this large outbreak of H5 infection in cattle, which is really unique. And whether that means that there's a higher risk for human adaptation, I can't say. But, as I said, it really scares me when an avian virus spends a lot of time replicating in a mammal. So I would say that there's certainly risk in the U.S. But these H5 infections are occurring in other parts of the world as well.
So, Arturo, you mentioned that you showed this really extraordinary study that a brief exposure to elevated temperature produces dramatic changes in a fungus, and those changes can be then maintained. Did I understand correctly that even
Erwin, you understood correctly. The fungi genomes, just like our genomes, are full of mobile elements. And when you raise the temperature, all these things begin to hop. The majority of the changes are deleterious. Most mutations are deleterious. But an occasional one will give you drug resistance, and another occasional one will increase virulence. So as the temperature goes up, you're not only going to have adaptation, but you're going to have new variants arise that could defeat our existing drugs, as well as give us diseases that we have no idea about because we have never seen them before.
It's interesting. This is a bit way off, but I've been interested through the years in some of the older studies on the temperatures of organs within the living body under different stages of metabolism. And it's really extraordinary. The liver in particular undergoes quite a wide range of temperature change, even though the systemic temperature hasn't changed that much. And I'm sort of curious whether the link between metabolism per se and heat and the change, particularly in fungus and bacteria, are they all linked in a sequential kind of way, or are these separate phenomena that we don't fully understand?
Yeah, I don't know if they're linked, Erwin D, but I think that one thing that you hit that I think is very important for the audience to realize is that when people say I'm 37, that's an average core temperature. You're absolutely right. The lung is a slightly different temperature than the liver, and certainly your skin. So you're a mosaic of temperatures. And that these temperatures we know, for example, that certain infections are both bacterial, like Mycobacterium marinum, only occur in the skin because the skin is cooler. The same thing with sporothrix. Some things just can't disseminate to the internal temperatures. But this is an area where there's been very little work. Primarily because, for a variety of reasons, but I think it's going to become increasingly important as humanity faces climate change.
So, Jeff, we have a question of whether flu viruses have been transmitted in domestic cats and dogs, and are there any cases of transmission to humans from cats and dogs?
Domestic cats have been infected with flu, including some fatal H5 infections recently in the U.S. Dogs actually have their own strains of influenza. There was a strain of canine influenza that emerged after adaptation of a horse strain of flu to dogs in Florida that then has spread and has been endemic. And then an independent emergence of a completely avian virus adapted to dogs in South Korea about a decade ago. So dogs clearly have been a host species of influenza, but transmission events between humans and dogs and between humans and cats is not something that's been widely observed.
So, Jeff, we have a couple of questions about Tamiflu and about immune serum and the treatment of influenza, or for that matter, I guess it applies also to COVID, to coronaviruses. You want to comment about both the Tamiflu with flu, and maybe you both have experiences or ideas about the helpful use of immune serum. So whomever.
Sure. Well, I can start, and Arturo could chime in. In terms of antivirals, with influenza, one of the problems is that the virus replicates so rapidly, and that it achieves very high levels of replication, in a sense, before you're symptomatic. So, in an ideal world, taking oseltamivir sort of as a prophylactic, like, for example, if you had a flu outbreak in a nursing home and you could immediately give oseltamivir to residents in the nursing home who would be at high risk of an influenza infection, I think it's really excellent. The problem is that if you wait more than a couple of days after the infection, and so by the time you're symptomatic and you wait 2 days, that's maybe 4 or 5 days of infection, it's often just a little too late. I mean, so the drug actually works, but you you're actually making the diagnosis and applying the treatment in a sense too late. So that's sort of the problem. In terms of immune serum therapy, certainly, obviously, for 100 plus years, that is very important. Obviously, antibodies derived against that neutralize or otherwise at least bind to and have other immune functions related to antibody binding to pathogens like viruses or other organisms. Obviously, clearly useful, but expensive and more invasive. So, that certainly there's a utility of that, but it's hard to have that be broadly used in a community outbreak.
Let me add something to that. During COVID, I was very involved in the use of convalescent plasma. And in the United States, 600,000 people were treated with convalescent plasma in the first year of the pandemic. And last year, we analyzed that it saved tens of thousands of lives. It was one of the sad things of what happened in 2020 was that the history of medicine had been forgotten. Antivirals, as Jeff just said, and antibodies work early in the course of disease. They don't work after a certain time. They're not going to work once you have a COVID lung full of inflammation or flu inflammation. But if we get hit again, and if we have another situation with one of these viruses, I think that we learn how to use it. And what is going to be critical is that we're going to have, there's probably not going to be small molecules to treat the whole population. But certainly, if you set up infusion centers, you need to treat people as an outpatient. You shouldn't be giving them a unit of convalescent plasma when they are in the ICU. It's not going to work there. But it is going to work for people at high risk that you basically get to early. And a lot of us have done a lot of thinking and planning that if we get into that situation, we will have learned from 2020. And I think we will be able to take an edge off until vaccines and monoclonals and things come on board.
So that gets into other questions that have been asked about. Jeff, maybe you want to respond to it. How do we respond globally? What is the machinery that's currently in operation to determine the emergence of a new strain, not necessarily influenza, but I suppose any microorganism? Where do we stand in our global society for detecting these early in the game before they've gotten on an airplane or something like that? Can you summarize that situation?
Well, it's always a challenge, and resources differ in different countries. They're not all the same. But in terms of influenza, there has been a long-term sort of global influenza surveillance network that was put together, in which you have national laboratories, but that they share data internationally and in general, because flus tends to circulate, again, in a sense relating to temperature in one sense. That this tends to be a winter month viruses. People tend to get respiratory viruses more in the winter months. So there are two winters each year, one in the northern hemisphere and one in the southern hemisphere. So the surveillance network and the vaccine strain selection for seasonal flu actually usually happens twice a year, once for vaccine selection for northern and southern hemispheres. And this is an international organization. The CDC in the United States plays a major role in this as one of the four major centers. But nations are able to make their own strain selections, and whether or not they want to have large-scale influenza vaccine programs. But in terms of the emergence of other viruses, this is a continual challenge, and that. If most of these viruses are coming from animals, either wild animals or domestic animals, the vast majority of the world's population are no longer living directly in contact with many of these animals that are the risk. And so it's often in more rural parts of the world, often in underdeveloped parts of the world, places in which there's expansion into less developed areas that are still more rural or wooded, where you're seeing this. So it is a problem, and as we know, these things do spread around, and that while the goal here is to think predominantly about what we can do to protect Americans, that these are international events, and we have to think of that in that regard. So what happens in another country can easily happen in the United States, as we've seen the importation of Ebola, or of course the SARS-CoV-2 as a fantastic example.
Thank you. Arturo, we have a colleague who asked, a dentist, or who asked whether anyone has looked into the correlation between the extent of fungal infection, mainly with C. albicans, a resident microbe in oral cavities, and temperature range in dentistry, that is, the formation of caries or denture-associated issues. Has any of this work with fungi sort of been taken up for investigation? And
so the Dental Institute has been very supportive of Candida research, obviously because of the problems that it can cause in the mouth. But to my knowledge, there has been no study, for example, of differences in temperature and and susceptibility to disease. One thing that I want the audience to know is that even though the humans are supposed to be 37 degrees, we are not all 37. There are some of us who go through life at 35, some of us who go through life at 38. And the mouth is no exception. I'm speaking to you now, the temperature there is going to be very different than as air is coming in and out than when I close it, when Jeff speaks. But again, the point that I'm getting at is that these very fundamental problems have really not been explored, to my knowledge, in any significant way.
Okay. So we have a provocative question here for you, Arturo. Can you envision a fungal pandemic of hunger caused by a fungus wiping out a food stock, such as, say, the potato blight of something like that? Is that something to worry about?
That has been a big worry for a long time. I mean, people have estimated that if, for example, fungi, there are rusts that can take out wheat, that can take out rice, that can take out corn. People have done the calculation that if we were to lose two of the major grains at the same time, humanity doesn't have enough calories. You simply cannot find enough calories in animals or in other way to survive for the 8 billion of us who's around. So yes, the U.S., the Department of Agriculture, and people have been very worried for a long time of the fungal threat to humanity through agriculture. I thought what you were going to ask was, can I envision a fungal pandemic of humans? Well, fungi are certainly pandemic for the frogs and for the salamanders and for the bats. I think the requirements would be that you would need to have a fungus that reproduces in the lungs and sporulates. And if it sporulates in the lung as you cough, you will then have human-to-human transmission. That doesn't happen among the known fungal pathogens. The known fungal pathogens are not communicable. But in the natural world, there is no reason for that. I mean, the fungi, go to your refrigerator, put some bread in, and you could just see the hyphae growing out. This is totally within the realm of possibility, and I certainly hope it doesn't happen. But could it happen? Sure. Just like when I went to medical school, I was taught that retroviruses did not cause human disease, and the coronaviruses only caused the sniffles. And since that time, we have had the HIV pandemic, and we now have 3 highly pathogenic coronaviruses. So I think we're warned.
Jeff, it's been proposed, and I guess there's some studies, quite a few, to draw some correlation between tropical jungles, particularly in sub-Saharan Africa, South America, and the increasing proximity of human habitation, tearing down the forest and so forth, and thereby bringing the high mutation rates of living microbes in the forest into contact with potential animal and other vectors, and sort of like the HIV story. Do you adhere to that concept, or do you have a different interpretation?
Obviously, certainly that, as Arturo elegantly pointed out, that the vast majority of viruses that are not already fully adapted to humans, that come from animals, are not just in the environment. They're in these animals, and so those animals are either agricultural domestic animals that people are exposed to, or wild animals. And that obviously in different environments people are exposed a lot. So I would assume that while there's been very little surveillance, that if you look at what I would call the human-animal interface, I bet that there would be a lot of viral chatter. That is, but what I mean by that is that the viruses are probably being passed back and forth between different animal species that interact all the time, and oftentimes maybe this is all just below the radar, that no productive infections occur, but occasionally you'll have one-off infections, and then occasionally you might see other things. So, this kind of surveillance at the human-animal interface is obviously critically important, but it's going to be extremely difficult to do.
Well, we only have a few minutes left, and of course, everybody listening, I'm sure, shares the not just the excitement, but the information, and more importantly, the challenge and opportunities, even for people who don't specifically work in these fields at this time, but maybe have technologies and ideas that would contribute very much. That's part of the purpose of this course. But I think it would be interesting to everyone listening, and perhaps each of you might share what you think is likely to happen next, or what progress, what steps would you like to see taken to help solve some of these long-standing problems. What turns you on as the biggest challenges at the moment? Arturo, you want to go first?
I don't know if these problems turn me on, but what I would say is, what I would like to know, I would like to see more basic research goes into adaptation to temperature. I think that, with things like today with AI, with AlphaFold and things like that, what we have learned is that there may be a lot we can learn and predict, or at least have some statistical likelihood of happening. What I would like to see is both microbiologists and immunologists to change the temperature in their experiments.
Jeff?
Well, I think those are great thoughts. I think that the challenges that we've seen in the control of respiratory viral infections that I try to explain, are real and remain, and not through lack of effort for decades of people trying extremely hard and spending their lives trying to do better at developing antivirals, other therapeutics, and vaccines to control such viruses as the coronaviruses and influenza viruses. So, but what it's going to take is a huge community effort, and so I'm a huge fan of multidisciplinary work. The virologists don't necessarily go to the meetings that the immunologists go to, or read the immunology journals, and people are so siloed, and people looking at other host factors and temperature and environmental issues, and there's so many variables that people work on their one little problem but don't think about the bigger issue. And of course, one person in one lab group can't look at all those things, but I just think that what we need to do is encourage much more collaboration and cooperation, especially across disciplines and fields, and especially encourage the young people in science to develop this kind of collaborative and cooperative attitude, because I think that's the only way we're really going to make progress, is by combining all these disciplines and sharing information and ideas so that better solutions for us can come out. I think the traditional models of people working in their labs on their one topic, while certainly satisfying and interesting, are less likely in this current age to yield the big results that you would get by a multidisciplinary kind of effort. So that would be my my thing that would turn me on the most.
You touched on, you raised the question of young people and training. And Arturo, you've given much thought and elegant writing to these issues. What would you like to see happen in terms of training, graduate, PhD, MD, whatever, to try and bridge this gap from that perspective?
Well, I second Jeff wanting more multidisciplinary studies. I will tell you one problem that we have. Our scientist training is too specialized. We basically take people and we train them to do one thing, and they're great at it. And we can train somebody in a couple of years to do deep work. We don't train them to be generalists. And in fact, fields frown on people that look into other areas. We need to change the culture of science such that people are to embrace looking at other questions. And we also need to preserve what is good in the current PhD system, which is to train people to do deep work in a short amount of time, but also think of giving them the tools so that they are scientists, so that when you present them with a problem, they don't say to you, that's not my area. What you want them to do is to embrace the other disciplines, because great progress is made at the interfaces. But most fields spend a lot of their energy killing off the interfaces.
Well,
But we're going to solve that. That just takes time, Erwin.
yeah. Well, like the two gentlemen on the Brooklyn Bridge,
Yes.
you two have certainly exchanged and presented not only very exciting information, but ideas that even go beyond the.
I have to order early.
Okay, but where are you ordering from?
Appreciate very much your being with us, and I know that I'm speaking not just for myself, but for all of the many people around the
She said we
world
probably need
who
early.
will
Otherwise, I have to
access,
wait till I get home.
or attended this session.
I won't be home till
So we
tomorrow.
want to thank you both very much for taking time from your very busy activities, and hope our paths cross soon. Thank you again.
Thanks,
Thank
everyone.
you. Thank
Thanks,
you,
Arturo.
Jeff.
Great to see everyone.
Bye,
Thank
everybody.
you. Have a good evening.